So welcome everybody. On behalf of the organizing committee, I'd like to thank you for joining us this afternoon or evening, wherever you are on the coast. So this is the second part to our 2024 Virtual Orchard Meetup Series. You can see on the screen here, we've got 4 sessions that we're holding over June. We started things last week. Talking about kind of trends in water availability. And now we're going to dive a little bit deeper today to talk about the role of water in tree physiology and quality. So we try to keep these as sort of informal and open as possible. So invite you to join in with the discussion whenever you'd like. If you can use the chat feature, that's going to be on the Zoom screen, that's going to be at the bottom of your screen. You'll see the chat option. If you can put your questions or comments in there, try to avoid, there may be a Q&A, try and avoid using that. Let's use the chat feature. If you prefer to be anonymous, then you're welcome to send questions, comments directly, perhaps to Bernadita or to Daniel. They're going to be kind of monitoring the chat. So, so if you see them, then you can send those questions directly and they can bring them forward. So let's see, before I get started though, we're gonna move to, let's go to the next screen here. I do just want to take a moment and acknowledge the team. There's both going to be some people leading the session today as well as working behind the scenes. So this series started 4 years ago, and Mario, you're welcome to give some background with this as well. But it's been kind of continuing to grow. I was welcomed into the group a couple of years ago, and I'm happy to have Canada be a part of this. But just to introduce the folks, my name is Kristy Grigg-McGuffin, and I work with the Ministry of Agriculture in Ontario, Canada. And my co-host today will be Emily Lavely, and she's with Michigan State University. We also, though, have Daniel Weber with Penn State, Caitlin Hengel and Lindsay King. They're both with Ministry of Agriculture in British Columbia. Mario Miranda-Sazo is with Cornell, and Bernadita Salado is with Washington State. So it's a great team to be bringing this together. So in terms of the kind of the format of these sessions, again, keeping it relatively informal, but we are going to start with 2 of our guest speakers tonight. And you're welcome to, if you've got questions that are specific to the presenter, you're welcome to enter them into the chat and we can address them following the speaker. Otherwise, we can kind of hold off until the end and have that during the kind of the open Q&A. But following our 2 guest speakers tonight, then we'll have an open discussion with a grower panel. They'll introduce themselves and we'll get into some questions with that coming up. So that's going to kind of be the way that things are going to go. We're hoping that we'll wrap things up by 8:30, but the way that our discussions often go, sometimes it can go a little bit longer. But we do hope that you're able to stay on with it to the end, because the discussion is always great. There's also going to be a survey link that's going to be put up near the end, and we do ask that you take a moment to complete that. So stay tuned. That'll go in the chat as well as onto the screen. So keep in mind with that. From the team, is there anything that I missed? All right, if not, let's move on. I'd like to introduce then our first speaker of the program. And so Dr. Alan Lakso, apologies, joins us from Cornell University. And so Dr. Lakso is or was a research and teaching professor at Cornell for more than 40 years, but now I'm sure he is happily enjoying retirement. He's still active, though, in research, focusing on apple fruit development and the physiological, environmental, and cultural factors that support or inhibit it. So Dr. Lakso, thank you for joining. I'm going to stop sharing and allow you to do that on your side.
Okay. All righty. Let me get the... Okay, does that show up okay? Great. Okay, well, thank you very much for the opportunity to talk to everybody. It's always good to talk with growers. And try to figure out what's going on and what we can maybe do about it. So I was asked to just talk briefly about some of the kind of fundamentals of water stress effects on tree physiology. And I'll primarily be talking about apples because that's what I've worked most with in the tree fruit area. But of course, a lot of the main responses are similar for other crops as well, other tree fruits. So um, when you think about water stress, it's a lot like economic stress. If your supply is insufficient for the demand, you're under stress. And the same happens with water moving through a tree. The demand is the drying, the air that is evaporating water from the leaves of the tree, and that's setting up a water potential gradient, like Ohm's law, where you have a gradient of potential. And as the water moves through, it is coming in from the supply side. So if you look at the system, simply, you really have your water supply, the rain, irrigation, and soil water reserves that you may have at the time, and then how the roots access that, and then the tree growth and physiology of the tree will affect how much demand there is also, and which is superimposed by the weather, which has a major effect too. So if we take a little closer look at this, you can see that there's a lot of different factors that are involved in the development of water stress in trees. Of course, you have the rain irrigation, but the soil type, the depth of the soil, has a big effect on how much water is available, the soil texture, and the actual water that's being held there. But the plant doesn't really sense the content of water. It senses the soil we call water potential, which is how hard it is to get water out of the soil. And then, of course, the root density and distribution has a big effect. You may have 6 feet of nice wet soil, but if the roots are only in the top 18 inches, you're still limited as to what the tree has available. And the thing about fruit trees is most of the trees, especially apples and grapes that I've worked a lot with, have very low root length densities. So in a volume of soil, any given volume of soil, there's really not very many apple roots. For example, if you have competing grass, there may be 25 to 200 times more grass roots in that same volume of soil. So the root density and distribution is a really important aspect, and the root density of our fruit trees is really quite low, and so that's one of the limiting factors that can lead to additional stress. So if we think about this, if the weather is a major factor that's involved, then you can see that there's actually for a given tree at a given time. Of course, the phenology, if you're very early in the spring and you have very few leaves, you won't develop as much stress as full canopy, say this time of year. But say at this time of year, we look at a plot, and the plot here we express in this water stress units, which are negative. So it goes down, and that they're negative because it's a water deficit, basically. And so the lower than that number, the more stress you have. So up near the top, there's very little stress, and then as you go down, it's more and more stress. So this is a plot from Terrence Robinson and Luis Gonzalez a couple of years ago, where we were monitoring water status in the trees in a trial where they had well-watered trees in the blue line, and then the red line were some that had been rain shielded and had developed some, if not extreme stress, but some water stress. And what you can see is that if we look at the blue, that's the well-watered tree, that around first thing in the morning, 6 o'clock in the morning or so, when it's coolest and most humid, you don't have very much water being used by the tree. And so it's able to absorb from the soil and reach maximum hydration. And then as you go through the day, the tree starts losing more and more water. And this is on a sunny day. And so you can see that water stress develops, even though you have good soil and water, develops because the atmosphere is just sucking a lot of water through the tree until later in the afternoon, when the temperature drops off and the sunlight decreases, and then it comes back up. And then first thing the next morning, it's pretty much the same, indicating the soil moisture is still quite good. Now, if you have rain or some type of rain shielding, in this case, where you're not getting as much soil moisture, you can see first thing in the morning, there's already some stress simply due to the soil just not being able to provide full hydration for the tree. And then it goes through the same sequence during the day as it goes down and then comes back up. And so you can see that you have some soil effect, but we also have a very strong weather effect. And that can also be seen on the right side then on a very cloudy day that just followed that sunny day, where there was a very, very little weather effect. And you can see it was mostly a soil moisture stress effect. So because of this combined effect of soil and weather, and then of course the phenology of the tree, how much leaf area you have, can lead to a pretty complex and dynamic situation of water stress. So water stress is almost never exactly constant. Okay. Alrighty. Oh, okay. Now oops. Try again here. It points out that, a drought is a meteorological term of just not as much rainfall as you expect. But drought stress is really what we're interested in. So the climate makes a huge difference. If you're in Washington, this is around Wenatchee, you've got very strong evaporative demands, bright sunshine, and in many cases you'll have a very high water use rate because of that, and so you can get into stress a lot quicker. So versus this is in New York. Just happen to have a picture of a vineyard, but it's just showing that we get a lot more cloudy days, more humid, and so a cool, humid climate, you can have a significant drought in terms of not getting the rain, but you may not necessarily have nearly as much drought stress. If you have a heavy soil, you may be able to go several weeks before the soil runs out of moisture. So now this is a plot just showing what the actual over, say, about a 3-week period, I guess it shows here, that water stress is going up and down every day. This is some data from actually in almonds from Ken Shackle at UC Davis, using the water potential sensor that's giving a continuous reading of the actual water stress in the plant. Again, more stress as you go down. And this is just showing at the beginning of that period, the values were much lower, indicating some soil moisture stress already was in that case. I should point out this dashed line is sort of the target level. You want it to be somewhere around 18 or 20 bars as the level to have the middle of the day, the lowest value each day. I'd like to be in that range. If it goes much lower, then that's a little bit too much stress. And so this is one where they're trying to maintain a stress level. And so you can see initially it was quite low, they irrigated, it came back up, and the top value is that soil moisture that supplies the tree. And so it did bring it back up, but you could see that there was still some decline in the water potential during the day, so needed a little more irrigation to get the soil moisture up. And then once after that, got the soil moisture to be fairly high, and the daily minimum value was around the target for most of the time, except for the last day or two before irrigation again. So this is the type of behavior of that system that we're dealing with. Okay. Now, I should point out the timing of stress is really important. In many ways, we're fortunate that with our tree fruits, and especially stone fruits, that we get a fairly early bud break in the spring. And also the canopy can develop quite quickly. And especially in the case of apple, by the time you're at bloom, you could have a canopy that's got 20 to 25% of the final leaf area, and maybe about a third of the amount of total sunlight interception going on. And that can be still quite early in the season. And that's when the fruit begin to develop. And as we'll talk about a little bit later, the early fruit growth by cell division is really critical for good fruit size. And that can be inhibited by water stress. But during the early spring, that soil moisture is usually higher in most cases, not always, but most cases we have replace the soil moisture over the winter. And so we're able to avoid the more severe stress until the middle of summer, which is past the most critical cell division phase of the fruit. So that timing is something that it's essentially a factor of perenniality, because we don't have to build a tree every year. We can just pop out some leaves and get going. So now I mentioned the development of the canopy is something that can be inhibited. This is some work by a student of mine. He looked at the shoot growth rate with water potentials and water stress. So in this case, on the right is no stress, and as you move left, it's more and more stress. So what we find is with shoot growth that we find a very kind of almost a linear drop off in the shoot growth rate with some stress, and it drops down quite quickly. So the development of additional shoots, extension shoots beyond the initial spur canopy is really quite sensitive to water stress, and it's because the shoot tips are always growing by cell division, and cell division is a very sensitive process. So as long as it's continually growing by cell division, it's going to be quite sensitive. But with apple, again, we have a spur canopy that comes out quickly. So it's not like we end up with no canopy if we have stress. We end up with a spur canopy, maybe not so many extension shoots. And the spur leaves have the ability to make it through the season and still be productive, which is quite unusual. So now I should point out, and this is a plot with looking at roots. I should point out that there's quite a strong interaction between cropping and water stress, and actually other stresses like pest stress and things like that. And this is a study where they looked at in some younger trees, where they looked at the number of roots that they could observe with different levels of water, from 150 and 25% of full water, whatever that is. If you had no fruit in the green bars, you can see that that dropping from 100 to 50 didn't really affect the roots very much. In some cases, you find the roots actually will grow more. Seems like there's a mechanism for, if you start running out of water, the plant will produce more roots to try to go get more water. In this case, it was pretty much the same. And then if it was too dry, then just the production of roots stopped. But in the red bars, these are trees with fruit on them. And even when there was high soil water, they didn't produce a lot of roots either. So we see this with a lot of interactions between stresses where they're interacting and getting a double whammy if you have two stresses at the same time. Okay. Now let's talk about fruit itself and effects. Just a quick review about this is the growth habit of apples, and this is some data that we took in Geneva. There's if you have a very good year without cold temperatures right at the end of the season, and it was a good season, warm and sunny, and we thinned the fruit to a very light crop because we didn't want to inhibit their growth. We wanted to see how they would grow just inherently if they weren't limited. And they grow as you know by cell division. Initially, it's an exponential kind of growth for about the first 4 or 5 weeks, roughly. Most of it by usually three weeks or so. But during that cell division period, it tends to be fairly sensitive to stress. I hope my little dog isn't too noisy in the back. Yeah. Any case, that's a sensitive period. And then during cell expansion, it's basically sort of a storage mode, and that is less sensitive to stresses. Now, it can slow down because of later in the season because of heavy crop load or additional, or colder conditions, those sorts of things. But they can grow almost linearly during that cell expansion phase if they're not limited. And I'll show some work about the growth effects. So in the cases, just a diagram, a little general diagram, just showing that if you get that inhibition early, even if you don't have any water stress the entire rest of the season, you've lost cell numbers, and you can end up with a significant reduction in fruit size. And that's seen here by that same student, where he had 2 different years where there was an earlier water stress, and this was a fairly mild stress. It wasn't severe. An earlier water stress, and then one that was later, not late, but just after cell division. So it's during cell division, just after. And you can see that if he had the early water stress during cell division, and then we watered the rest of the season, you can see that there was lost fruit size due to the cell numbers, and ended up having a significant reduction in final fruit weight, where when there was a mild stress occurring just after cell division, it did show some decline, but then eventually it seemed to be able to just gradually catch up, and there wasn't really a significant effect later. There may have been a tiny crop load effect. So you can see that that the timing is quite important. Now, when we actually look then at the relationship of fruit growth to that water stress potential, you can see I got the shoot growth I showed earlier, and I superimposed that green line from the shoot growth onto the fruit growth plot, and you can see that there is a little bit of a resistance or tolerance of water stress with fruit growth initially, and this is primarily after cell division. So there is a little bit of ability to tolerate some mild stress before it starts declining. And in some cases, the difference in, say at a value when the shoots have basically stopped, you may still have something like 50 to 80% of the maximum fruit growth rate. And if you have large fruited variety, you may be able to tolerate that, and the water stress would, if you can regulate that, would control vegetative growth as well. So now this is a plot just showing from Bob Ebel in Washington, in Yakima, I believe, where it looked at the fruit weight, and they stopped irrigation at different dates. And so this bottom one is no irrigation, and they stopped June 1, June 15th, 29th, then August and September. And you can see that it's really a cumulative effect. If they stopped earlier, it accumulates more stress, and you end up with the reduction in the final fruit size. This was also looked at where they looked at using a regulated deficit irrigation, and they did that until about mid-August or so, with the arrow here. And you can see with different crop levels. So with the light crop load versus the no RDA, you can see that there was an effect initially, but it looks like a little bit of that caught up. So it wasn't a very strong effect. But if you started getting into moderate or heavy crop during that RDI period, they really shut down their growth rate quite a bit. And then even after the rewatering, you could see the growth rate was pretty much similar to the others, but they had just lost so much earlier in that period just before the RDI was stopped. So this shows that crop load effect as well. And just one last set of plots here, again from Bob Ebel and Probsting, showing the fruit, the harvest weight of the fruit related to they used a cumulative water stress, which is basically the water, that water stress value multiplied by time, number of days. So it's pretty much like degree days, except it's water deficit days. And so they were able to use that as an accumulative way of looking at the effects on the final fruit weight. And you can see it correlates very, very highly to that cumulative water deficit effect. And then also point out stress in year one, it's a perennial plant and it's producing that flower bud development. So it can have major effects on the yield in year two. And so they looked at the water deficit index, the same index in 1 year, and then the second year, you could see a quite dramatic drop off in the yield the previous year. And we can see that actually with European red mite damage, that it can have significant effects on the cropping from the next year. So in any case, I hope that gives a little bit of an overview, and looking forward to Lailiang's talk and further discussion. Okay.
Wonderful. Thank you very much, Alan.
Okay.
Are there any questions? Daniel, I believe there was one in the chat.
There was. Gil, did you want to ask it yourself, or do you want me to just read it for you? It's under your call. You can unmute yourself if you wish. Okay, so the question was, could implementing a dynamic maximum allowable depletion with phases of lower stress during the reproductive physiology improve yields?
Yeah, actually, I mean, the whole goal of irrigation really is not to replace the water, but is to get the plant to have the optimum water status. And so since it's a very dynamic process going on during the season, if you can monitor that and understand what the effects are and what might be acceptable level of stress, before having a significant reduction of growth, then having a dynamic, in your case, you're talking about depletion of the soil water, I assume. Okay.
I'll keep my eye on the chat to see if he has any follow-ons, Alan.
Okay.
Oh, yeah, correct. I've been implementing a static 40% MAD. What would be a target in reducing this stress?
Well, actually, we find when we really take a lot of data and look at the relationship between the tree stress and the soil moisture stress, a lot of cases we don't see very good relationships. Now it can be better relationships in drier arid zones, where if you're drip irrigating, there have a moist zone where you're irrigating, and then a lot of the rest of the soil is quite dry, and so the roots will concentrate over into that drip zone, and so you have a better idea of what the roots are actually exposed to. In the east and in central Michigan and more humid climates, we find that the roots go all over the place, and so we don't always know where they are. So we don't always have a very good reading of what the relationship is between the soil moisture and the tree stress. So which is why for years I did a lot of measurements with this is called a pressure chamber, to actually measure the tree itself. But that's another whole long thing that probably should wait. Okay.
We do have one other question here. Can't quite read what the name of the person is with the blue on the black there, but it says, what about stress from overwatering or overmoist roots, and how can it be corrected to avoid losing trees? Any recommended readings on this? Okay, and it was Claudia who submitted that.
Okay, well, we need to realize that that overwatering or flooding stress is not water stress, it's oxygen stress. And so it's kind of hard to use water as a proxy. Too much water is causing the oxygen stress, but I think the amount of oxygen stress will depend on the soil texture that you've got. Basically, I don't know. I really didn't deal that much with, flooding or excessive moisture. One of the things that does happen is that you basically asphyxiate the roots and they die. And then you have, when it does turn warm and dry, you end up with a root system that's been debilitated and it doesn't have the ability to take up water as fast. So what you may see is the low spot where it's been flooding or heavy soil. When it starts getting dry, they'll have a lot more moisture than a drier section, maybe a little higher. But the ones in the soil that have a lot of water will actually be showing drought stress more. They'll be wilting, not because they don't have the water, but because they've killed the roots. And the roots can't take up enough water to supply. So that becomes an internal resistance in the system. So in any case, certainly, having a good drainage soil, we use a lot of drain tile in our area. So just making sure there's good drainage, and ultimately, if you can, be sure that you don't use excessive water. But I'm sure others have more experience with excessive moisture than I do. So, okay.
I think that's all the questions for now, Kristy.
Excellent. Thank you again, Alan. Much appreciated. And Alan will be around for the remaining story.
Yep.
The joys of home. So the next speaker I would like to introduce is Dr. Lailiang Cheng.
Are we doing the poll?
Do we want to do the poll after the Lailiang?
Oh, okay. I'm sorry. I thought you wanted to do it in between. I forgot. That's on me. Sorry about that.
Not a problem. My dog will just continue barking. So, I would like to introduce Dr. Lailiang Cheng, and he joins us from Cornell as well. And Dr. Cheng leads Cornell's program in nutrient management of fruit crops. So thank you very much, Dr. Cheng.
Thank you, Kristy. Good afternoon, everyone. Let me share my slides. Are my slides good?
Good.
Okay. Ellen just provided an excellent overview of tree physiology in relation to water. And today, what I'm going to do is to talk about water supply and apple fruit quality with a special reference to Honeycrisp bitter pit. And Dr. Lee Kelsitz at Washington State University, and I put some slides together. I should be able to go over them fairly quickly. The effects of water on fruit quality can be broadly categorized into direct effects and indirect effects. So water supply can directly affect fruit developmental processes, such as cell division and cell expansion, as Ellen talked about. Therefore, final fruit size is affected by water supply. Water supply can also indirectly affect fruit quality through altering other processes, such as canopy photosynthesis and carbon supply to fruit growth, and nutrient uptake by the roots, and their partitioning to fruit. So my talk today is going to be largely focused on how water might affect root nutrient uptake and partitioning, sort of indirectly affecting fruit quality, particularly on bitter pit in Honeycrisp. Now, all the essential nutrients required for tree growth and development fall into two groups. One group is called phloem mobile nutrients. So these nutrients can be transported in the phloem as well as in the xylem. The nutrients such as nitrogen, potassium, phosphorus, magnesium, sulfur, boron, all of these are phloem mobile nutrients. The other group is called phloem amobile nutrients. These nutrients include calcium, manganese, zinc, copper, iron. Calcium is the best representative of this group of phloem amobile nutrients. In other words, these nutrients can only be transported in the xylem. So this figure shows you the calcium accumulation in shoots and leaves here, the upside down triangle, versus in fruit. For phloem amobile nutrients, such as calcium, the characteristic developmental pattern is that most of the nutrients gonna accumulate in shoots and leaves. Only a fraction gets accumulated in the fruit. So in gala fruit, at fruit harvest, by fruit harvest, about 86% of the calcium is in shoots and leaves, with the balance 14% in fruit. So that's a very acute distribution. Like leaves, shoots and leaves have most of the calcium, whereas fruit only gets a fraction of the total calcium that is transported from the roots to the above ground portion. This is because the distribution or the allocation of calcium and other phloem amobile nutrients is driven by transpiration only. So as we all know, shoots and leaves have much higher transpiration rates than fruit. So shoots and leaves Again, most of the calcium with only a fraction going into fruit. There's varietal difference though. We look at the Honeycrisp fruit on 4 different rootstocks. Just want to show you some of the data on G11. As you can see here, this acute sort of distribution of calcium between shoots and leaves and fruit is similar to what we have described in Gala. However, Honeycrisp fruit only gets about 6% of all the calcium transported from the roots to the above ground, with 94% of the calcium found in shoots and leaves. Because of this, Honeycrisp is more susceptible to bitter pit. So we compared Honeycrisp fruits with Gala fruits, and Honeycrisp fruits only have about half of the calcium found in Gala. So this is calcium. Now for phloem mobile nutrients. Oh, before we get to that, I just want to zoom in on the fruit accumulation patterns in fruit. Okay, in Gala and Honeycrisp. If we zoom in, we're going to find that for Gala, calcium accumulation occurs pretty much throughout fruit development. See that? This is bloom. This is end of shoot growth. This is rapid fruit expansion here, all the way to fruit harvest. Okay. For Honeycrisp, however, most of the calcium accumulation occurs in the first sort of 6 weeks after bloom, roughly. Okay. This is bloom, about 40 days after bud break. Another 40 days, that's pretty much it. And then after that, of course, Honeycrisp trees stop their shoot growth earlier than Gala. So by this point, shoot growth is pretty much stopped. And then after that, you don't see much increase in calcium in the fruit. This is largely due to the loss of xylem function in the Honeycrisp fruit. In contrast, Gala, the xylem functions really well throughout fruit development. That's one of the reasons why Gala is much less susceptible to bitter pit than Honeycrisp. Now let's look at the phloem mobile nutrients. Potassium is a good representative, a good example. For potassium, let's say, if we look at the potassium accumulation in shoots and leaves here, represented by the upside down triangle, versus the potassium accumulation in fruit, represented by the open circle. It's very clear potassium accumulation in shoots and leaves and in fruit is demand driven. So when the shoot growth stops, potassium accumulation pretty much stops, remains unchanged from end of shoot growth all the way to fruit harvest. For fruit, Ellen described that apple fruit growth, if there's no resource limitation, it follows in an expolinear pattern. So the initial growth is exponential, followed by a linear increase all the way to fruit harvest. So potassium accumulation in the fruit follows an expolinear pattern as well. At fruit harvest, about 70% all the potassium in the new growth. This represents all the potassium in the new growth, which is the potassium in shoots and leaves plus the potassium in fruit. So 70% of the potassium in the new growth is found in fruit. The balance 29% is in shoots and leaves. For Honeycrisp, very similar patterns again, but the fruit accumulates more potassium than Gala. This is about 78%. And these trees have actually lower potassium status. You know, leaf potassium level is around 1%. For Gala, the leaf potassium level is around 1.6%, but most of the extra potassium is largely found in the fruit peel. Flesh slightly higher, but not a whole lot. Now, for bitter pit, we all know it's a calcium-related disorder. But from the physiological standpoint, it is the calcium that is in the cell membrane that determines whether a fruit develops bitter pit or not. We don't have enough calcium in the plasma membrane. The cell membrane loses its integrity, as shown here. Basically, calcium binds to the negatively charged groups, like proteins, carboxylate groups from proteins, and phosphate groups from phospholipids, so stabilizes the membrane. We don't have enough calcium in the cell membrane. The membrane gets detached from the cell wall and then gets degraded. And that leads to cell death. When there is enough cell death, when there's enough numbers of cells that die, it is manifested as sunken pits. That's what bitter pit is all about. So because it is the amount of calcium in the cell membrane that determines whether fruit develops bitter pit or not, we need to look at all the processes that leads to the amount of calcium in the cell membrane. So calcium has to be taken up by the roots along with other nutrients first. That requires water. Once these nutrients are taken up, it is partitioned between leaves, shoots and leaves, and fruit. For calcium, that partitioning is strictly determined by transpiration rates, okay? But for the phloem-mobile nutrients, potassium, magnesium, and nitrogen, they can enter the fruit through the xylem, but they can also enter the fruit through the phloem. That's why in Honeycrisp, although the xylem function stops very early at about 6 weeks after bloom, the fruit continues to get potassium from the xylem, from the phloem, I'm sorry. So when the carbohydrates, sucrose and sorbitol are transported in the phloem with water to the fruit, potassium goes along with the carbohydrates. So potassium enters into the fruit primarily through the phloem when the xylem function pretty much stops at 6 weeks after bloom. Once the calcium is transporting into the fruit, there is a partitioning, there's a transport from fruit stem end to the calyx end. So if you compare Honeycrisp with Gala, you're going to find that at 2.5 weeks after bloom, not much difference. Yeah, Gala does a little better here, but at 7 weeks after bloom, Gala is still, the xylem is still very strong. Basically, we use the water-soluble dye called acid fuchsin. That comes into the fruit with water. So if the xylem is functional, the tissue gonna become red. But for Honeycrisp, at 7 weeks after bloom, you only see a little bit of xylem function. And this corresponds to this the plateau of calcium accumulation in Honeycrisp after 6 weeks, after bloom very well, okay? And based on this, we designed a diagnostic method called fruit peel sap analysis. Basically, we take fruit samples at about 8 to 9 weeks after bloom, when the fruits are about 55 to 60 gram size, to predict bitter pit incidence. But before we get into that, you may wonder, well, calcium, you said calcium in the cell membrane is very important for cell membrane function. Why potassium is also important? Well, potassium has two effects. If you have too much potassium, less calcium will get into the fruit. That's the first effect. Second, when the fruit has a lot of potassium, that will make the calcium in the fruit less effective in getting onto the membrane, binding those, proteins and phospholipids, as you can see here. So we basically compared Honeycrisp fruits with the bitter pit, okay, with the bitter pit, BP, but it's the healthy tissue on a fruit with bitter pit, and the pitted tissue. Compare the values of mineral nutrients with bitter pit free fruit. As you can see here, we measure the calcium concentration in the cell wall. That's where the cell membrane is bathed in, okay? So if we measure calcium concentrations in the cell wall space, there's no difference. But the fruit with the bitter pit have significantly higher potassium level in the cell wall space, which is also called the apoplast, okay? And as a result, the potassium to calcium ratio is significantly higher in the fruit with bitter pit than the fruit without bitter pit. So now come back to this pure sap analysis. So we take fruit samples at about 8 to 9 weeks after bloom, 30 fruits per block, get 2 pieces of peel tissue from each fruit, a total of 60 pieces of fruit peel tissues, and then we freeze them in a regular kitchen freezer overnight, and then let them thaw, use a garlic press to get the sap. We directly analyze the pure sap for mineral nutrients. As you can see here, bitter pit incidence measured by the passive method. Basically, we take 100 fruits at about 3.5 weeks before harvest. We leave them at room temperature, and then count the number of fruits that will develop a bitter pit in 3 weeks. Okay, so that's called the passive method. And we'll also assess the bitter pit incidence at fruit harvest. And then we take 100 fruits free of bitter pit, put them into cold storage, evaluate the bitter pit incidence after 3 months of cold storage. And then the total bitter pit incidence is the bitter pit incidence combined here at harvest and after cold storage. As you can see here, bitter pit incidence shows the exponential relationship with the pure sap potassium to calcium ratio. With about 2/3 of the variability in bitter pit incidence explained by the pure sap potassium to calcium ratio. Considering that there's still 2 months to go, we're very happy with the prediction power of this method. One of the reasons that this method works so well is that with Honeycrisp, most of the calcium accumulation in the fruit happens in the first 6 weeks after bloom. So we designed this diagnostic method at about 8 to 9 weeks after bloom. So by that time, most of the calcium has already got in. And potassium plays a role into this later, but we're going to talk about that, how water stress affects that a little bit here. Weather patterns and irrigation affect bitter pit incidence. So drought stress early in the season, typically from bloom to about 6 to 8 weeks after bloom, primarily during cell division. Remember, cell division is still the period where most of the calcium gets into the fruit for Honeycrisp. So if you have drought stress, you're gonna reduce fruit size by reducing cell number, you're also going to reduce the amount of calcium getting into the fruit. So that's why early water stress increases the risk of bitter pit. If you have a wet year, or you apply too much water, you do over irrigation, particularly from mid-season on, during the cell expansion phase of fruit growth, you're gonna make the cells so big, you're gonna dilute the calcium concentration, you're gonna bring more potassium into the fruit, leading to a higher potassium to calcium ratio. As a result, you're going to have a higher bitter pit risk. And this is some data my colleague, Dr. Lee Kelsis, collected in Central Washington. As you can see here on the left, bitter pit incidence is linearly related to fruit size. If you over irrigate, Honeycrisp is a large fruited variety to begin with, you can get pumpkin-sized fruit, as shown here. So this is Lee's son, Blake. Look at the size of that fruit. And it's not surprising that that fruit has a bitter pit because it's too big. When a fruit is too big, that again, it dilutes the calcium concentration. It brings more potassium into the fruit along with the water. So you end up with an imbalance between calcium and potassium. That's interesting. Why there's, okay, here. So Lee did a deficit irrigation in Washington on 4 to 6, I think 4th and 5th leaves Honeycrisp over 2 years. Basically, he set up, he had the full irrigation as control. And then at the early stage of fruit development, 15 days to 45 days after bloom, and then mid-season, 45 to 75 days after full bloom, and late season, 75 to 105 days after full bloom. So withhold water to about 50% of the, roughly 50% of the evapotranspiration, 50–60% for a month. And then he measured bitter pit incidence. 2 years, not very consistent, but if you look at the data in 2017, you're gonna see that the full irrigation control had high levels of bitter pit. Early season water deficit, essentially drought stress during cell division, further increases bitter pit incidence. However, if you control irrigation to create some water deficit, that reduces bitter pit incidence. And that's one thing Terrence and I observed in New York, like on years where you have a water stress early in the season, typically the fruits will have higher bitter pit incidence. If you have a moderate drought stress, let's say from late July or early August for about month and a half, that's gonna really help with reduction of bitter pit incidence. So here are the take-homes from my talk. First, water affects fruit nutrient balance as well as fruit size. For small fruited varieties such as Gala, Empire, adequate water supply throughout fruit development is critical for fruit size. Although more towards the, let's say, a couple weeks before fruit harvest, a little bit of water stress I think will be helpful for color development and sugar accumulation. However, for large fruited varieties that are susceptible to bitter pit, such as Honeycrisp, adequate water supply in the early season during cell division, but limiting water supply in the late season is very important for mitigating bitter pit. For young trees in low crop load situations, limiting water starting from the mid-season on is even more important, because for young trees and trees with low crop load, fruits tend to grow bigger. So by using water as a tool, you can sort of limit the fruit size and limit the amount of potassium getting into the fruit from mid-season on. That can really help with bitter pit mitigation. I think that's all I have. I'm sorry that it took a few minutes longer than originally planned. Thank you very much.
Thank you very much. That was wonderful. Have there been any questions that have come through in the chat there, Daniel?
I do not see any at this time, but
Okay,
if anybody has some, please submit them as soon as possible. One just did come in. Thank you, Joseph. It says, any information about water stress in Fuji?
Fuji, we don't have a lot of Fujis. Fuji, once we get into in New York, once we get into, let's say, October, we're gonna have more rain. So really, it's not a question I can answer well. I think perhaps Bernadita in WSU can answer this. Bernadita?
No, I don't have much information. We will have, because we have a new project here with a deficit irrigation on Fuji, so we hope to share that information later. But I guess what Alan Lakso mentioned, all apple varieties, I think we have to be cautious in that early stage to prevent a deficit in those stages.
I might make a comment sort of related to this, was that we did studies where we looked at the effects of European red mite injury, foliar injury. And what we found was that for us, the red mites would usually explode around the 1st of August or so. And when they got to high values, it really shut down the growth of the fruit. So we had a reduction in final fruit size at harvest. But when we did the nutrient values in them, we found that the smaller fruit that had been inhibited had higher calcium concentration, but per fruit it was the same. So it developed up until about that time, and then we just didn't dilute it with additional growth. Then post-harvest behavior was nice, except the fruit was small. But but there's that sort of dilution that can be due to water, but it can also be due to less carbohydrates going into the fruit. So that was kind of a odd thing that I didn't really understand at the time. But
thank you, Ellen, for that comment. Yeah, that's whenever you sort of reduce fruit size, you're gonna sort of mitigate the calcium issue. It's just less dilution effect, and that's the sort of when you control water supply mid-season on. With Honeycrisp, it's large fruited variety. As long as you control crop load really well, sizing the fruit is typically not a problem. So sometimes the fruits get too big. That can become a problem. So the larger the fruit, the lower the calcium concentration will be. And typically those fruits have more potassium, because whenever the water comes in, the potassium is going to come in. Because although the xylem function already stopped, but the potassium comes into the fruit from the phloem.
We have another question that was submitted by Rebecca. It says, how much deficit do you recommend in the later stage?
That's a good question. I think in Lee's experiment, he used about, like, if you use soil field capacity. And he said the control trees are at about 90%, 85, 90% of the field capacity. Okay, so those trees received 4 30-minute irrigations per day during the summer, during fruit growth. Okay, and for the deficit irrigation treatment at different stages of fruit development, they are pretty much the same. They used about more like 40 to 50% of the field capacity. So that's if you use that. And then I guess you can also use evapotranspiration. Like you have 100% ET, let's say that's the control, and then you can have, let's say, about 60%. Ellen, is 60% ET a sort of a more moderate, like 2/3 or 6%? Is that a moderate stress, or that's too severe a stress?
I don't know. I don't have good correlation between the percent of ET, because a lot of the ET of an apple is not quite the same as the standard
Yes.
ET we found because of some funny behavior of the stomates on pores on the leaves. But of course, it's not a constant value. You down to 50% of ET. It's going to take time for that to take an effect, unless you're in a very, very warm, dry climate with a small soil water reserve, so that you really have them on a string with the irrigation.
Yes.
Then you could get the effect pretty quickly. Otherwise, you're going to have some soil moisture, and it's going to take a while for that to get down to that 50%.
Yeah.
So it'll be kind of a gradual effect. In a lot of ways, of course, I'm not a large grower, so I know there's a lot of logistics involved. But at that point, if it's that critical, I would have somebody mark some fruit and actually monitor, go out and measure the fruit, and let the fruit tell you if it's enough stress to reduce their growth.
Yeah, I think when you apply, I don't know if we answered the question or not, but when you apply deficit irrigation, it's very important to make sure you have good evidence showing that the fruit growth is not affected too much. Because fruit size, if you apply deficit irrigation too early or too much, you're going to affect fruit size. For Honeycrisp, typically it's not a big issue, but for other varieties, it could be a big issue, because fruit size, apples are sold on size and color, essentially.
Yeah, as I pointed out on the effect of the crop load, and that is there's interactions, and if you're already struggling a bit for size because of a heavy crop load, you have to be really careful with trying to induce any kind of water stress. So really, the heavy cropping blocks really need to be monitored. Same thing for like foliar pests.
Yes.
That the heavy cropping trees actually needed a less severe threshold for mite control, and the light cropping trees that could tolerate more. So it's not a simple system.
I'm just going to jump in there and wondering if maybe we could pull in the grower panel at this time and then continue on with that discussion. It's great. This is great that's happening. So it'd be wonderful to bring in just a few more folks. I think, Emily, just for the sake of time, I think we'll skip over the poll and just go straight to the growers. So I'm going to hand things over to you.
All right, thank you. Yeah, is it maybe we could put the poll at the end or something if folks want to contribute. It's not competing with the survey, but thank you so much, Dr. Lakso and Dr. Cheng, for sharing that information with us and getting us thinking about these issues. So I see the grower panelists have put on your cameras. That's great. Right now, if you wouldn't mind just introducing yourself briefly again, sharing a little bit about your operation and tell us where you're from and what conditions you've been experiencing related to water so far this season. Kevin, would you like to go first?
Sure. My name is Kevin Bittner from Bittner Singer Orchards, a 400-acre fruit farm on the south shore of Lake Ontario in western New York. So far, I grow a lot of apples, peaches, cherries, prunes, plums, apricots. So far this year, our water resources have been adequate from rains. We have all our irrigation, our trickle irrigation all ready to go. About a third of our farm is trickle irrigation, all using municipal water sources. So far, we've had enough water. The temperature and the other weather has been kind of variable.
All right, thank you. Brian, would you like to go next?
Sure. So I'm on the north shores of Lake Erie in southwestern Ontario. We farm about 400 acres, 250 acres of orchard. And of the 250 acres of orchard, 200 acres or so have drip irrigation. That's tart cherries, apples, peaches, the whole gamut. The whole farm is systematically tiled. I didn't mention that last week. So we do have multiple microclimates on our farm and soil types, but we still tile the majority of our farms to allow us to control that comments about O2 deficits and stuff like that. By getting the water out of that ground and away from the tree, it seems to help the trees quite a bit. So, yeah.
All right, thank you. Aaron?
Yeah, so we farm a little over 2,000 acres here in Mattawa, Washington, just kind of located in the Columbia Basin in central Washington. And we're farming in a desert. So yeah, irrigation is very important to us for a lot of different reasons. Not just watering the tree, but sunburn control and taking stress off of the trees as well during the summer. So it gets pretty hot out here. We'll several days out of the year be 105 plus. So it's kind of an early district. We have silty, sandy, loam soils, so pretty good drainage. And I don't know if apples, blueberries, cherries. Most of that is apples. About 80% of it's organic. So we have a real focus in that direction. Did I cover everything?
Yeah, thank you. All right, and Jamie.
Yeah, hello. I'm from River Ridge in Michigan. We're on the west side of Michigan, so close to Lake Michigan. About 1,200 acres of tree fruit, mostly apples, getting larger into sweet cherries. Most of the new plantings all have drip irrigation and tiled drainage systems in them. For the most part, we've had pretty good spring so far. We've had the rain when we needed it, probably not too much. But it sounds like based on all the numbers there, we're in that period where we could use a little deficit probably. So everything's looking good so far.
All right, thank you all. I appreciate you just kind of sharing your experiences. I know I've been thinking a lot about this season compared to last season, which feels really different in Michigan. It was really dry last year, so it's interesting to just see how the trees are growing with so much water this spring. So that was another thing that I wanted to see if you guys had some thoughts on, if you'd like to share how you
Looks like we might have lost Emily a little bit there.
am I back
That was
now?
extraordinarily bad timing. Oh, okay, there you are.
Okay, I
You're
don't know what
back.
happened, but just
The joys of spotty connection.
the season or excess water Kristy,
You
you
do-
might have to take it from here.
That's no problem. You just came back. Not sure what you were going to be approaching with, but maybe we can kind of just talk a little bit in terms of, I mean, quite a few touched on the challenges, but if maybe you can kind of go into a little bit more detail in terms of what you face and maybe some of the strategies that you're implementing to try and manage those challenges. I think maybe can we jump back? We can kind of go through, maybe Kevin, if you want to kick things off.
Our biggest challenge right now with water is trying to make our best educated guess on how to efficiently use our water. It's a plentiful resource for us, and completely on municipal water. We've been using the newer models with we have a weather station right on the farm with a evapotranspiration calculator through Cornell. We've been using that to try and determine how much water we need at any given time and looking at the weather forecast. If throughout the growing season, if we don't have an inch of rain per week, we figure we need to irrigate. More recently, we're adopting the floor pulse microtensiometers. Last year we had 4 of those on the farm. This year I have 10. We're trying them just to get a better educated guess on how much water we need and how to better develop the models to predict how much water. We haven't automated any of our irrigation yet, but as these systems develop, I think that's in our future. One of our biggest challenges is we'll have a 40-acre farm all irrigated, but we can't get a large enough meter to irrigate all of it at one time. So we'll put it in different zones and rotate throughout the day to each zone 2 or 3 hours at a time.
That's great. Thank you. Brian, do you have any comments in terms of your challenges and strategies?
So we're very fortunate, and actually I'm going to give a shout out to Rebecca Short, who's on here. I see. She did a project a few years ago on our farm, and it really opened our eyes on our assumptions about that we were irrigating enough. And so those assumptions, because we do have multiple soil types, you think because you've irrigated for 2 hours or you've irrigated for 4 hours, you've put enough water in. And those assumptions got thrown out pretty darn quick, especially on our fox gravels and other sharper soils. And so we kind of had to readjust our mindsets quite a bit. Since 2019, I can name dates when we've had the feast or famines of rains, right? Like where we've had 5 inches of rain on the whole farm in 30 minutes. And then other times when we haven't seen 5 inches of rain in 6 weeks, or we haven't seen 5 inches of rain at all. So that's one of the things we've had to learn how to compensate for that feast and famine cycle of our rains, and learn how to properly gauge what's going on in the orchard using simple tools. Everything from a baking sheet to see what a transpiration looks like or evaporation looks like, to digging up soil with a shovel to make sure that the water cone's spreading the way you want it to from underneath the tree. So yeah, and we've changed some of even our drip irrigation structures to help them work better underneath our trees, like hanging them versus putting them right on the soil. That's really made a difference, actually, is hanging them. So yeah.
Great. Thank you. And Erin? Oh, sorry.
Can I ask if you could elaborate a little bit on what the difference is between hanging and putting it on the soil, since you mentioned that?
Yeah, Emily. So what we do is now in our apple orchards where we have wire, we actually suspend the drip lines about 18 inches off the ground. We've done that in apples and pears. Where we have it on the ground, which is under our tender fruits, because we don't have wire in those. We have them on the ground, and what I've noticed there is we aren't able to get a nice linear stretch of our water on the ground. Sorry, I talk with my hands. So we're not able to get that water to stretch along the whole tree line, which is what we kind of I like to see anyway, in the apples and pears that we're getting. Because pears require a lot more water than a lot of the other fruits that we grow. So we like to see that spread of the water.
Great, thank you.
Aaron, do you have any comments from your area?
Yeah, I think we kind of run 3 different systems. Everything's got drip line underneath the tree, and that's for our main irrigation and fertigation. That's kind of how we do probably, most of our watering during the season. We've got another valve and submain set up and in our blocks for frost control. It's under tree sprinklers for frost control early in the spring, and maybe we'll water some cover crop with that during the year as well. And then we've got our overhead cooling system to keep the trees cool during the summer. And I think that we really don't use the under tree that often. It's not really a huge issue for us. Fertigation and irrigation, we just have really light soils, and it's hard to over irrigate here. But there is such a thing as being, a good steward of the resource that we do have. But I think where we really get tripped up a lot is probably in overhead cooling during the summer. That's where we use the bulk of the water that we buy and we do cycle it. So we were 20 minutes on, 20 minutes off throughout the day. But some of our older systems, they're Nelson R2000s, which is a great sprinkler, but it puts out a lot of water. And so some of that's like 45 gallons per minute per acre overhead. And you're running that, for accumulation of, 4 or 5 hours per day. And so that starts to get things pretty wet. And so we've got I talk with my hands too, Brian. We've got some slopes that we're on, and almost everything is just a gradual slope here. And so when you're cycling, when you turn it off, everything goes down to the bottom and drains out everything on that last sprinkler at the end of the row. And then as soon as it's all drained, 15 minutes later, you're starting to back up again. And so you just keep doing this throughout the day. And we do tend to actually overwater at the end of those rows. And that's kind of not much of an issue anymore. Most everything we've updated now, we're using very fine micro sprinklers above tree. So maybe like 25 gallons per minute. So we're almost cutting that in half. And then we're putting check valves, drain checks, so they won't leak. And so we're actually starting to need to irrigate more during our summer cooling, because not as much is hitting the ground, which is great. But then, we're able to do things like what Dr. Cheng was talking about with Honeycrisp and doing some drought stress, because we couldn't do that before with all that water hitting the ground during that time. So I guess main point, cooling is a big thing for us, and it's caused, excuse me, it's caused some problems over the years for us with overwatering. But I feel like due to technology and the investments we've made, we've kind of gotten away from that, and not the issue it used to be. So that's all I got.
Kristy, if you don't mind, I have a couple of questions that are related to what Aaron said to Lailiang. Aaron, you mentioned this water that accumulated more in some areas. Could you relate that into fruit quality? Did you have more issues with bitter pit, for example, or other issues in those areas in the past, or you were not able to distinguish those?
I guess the things that we noticed the most, and I think this is what Dr. Lakso was talking about, was that, the water would actually pool there, and it would get smelly, and you could tell it was anaerobic, and there's no oxygen down to the roots. So you could see the trees starting to decline. They didn't necessarily die. And what's interesting is that fruit always ripened about 7 to 10 days earlier than the rest of the block, because I don't know the exact mechanism. Someone smarter can talk about that. But we would always notice that, yeah, that fruit matured about a week early. We'd go pick it. It was smaller, and you could see the trees, they were just really unhappy. You could see chlorosis, and they lived, but there's just disease-ridden soil underneath there. And they've snapped out of it since we fixed the problem, and things have gotten better. But during those years with standing water out in the block for a while. Yeah, just early maturity, smaller fruit trees were pretty sad looking.
I may just want to add that, Aaron, the early ripening may have something to do with the increased ethylene produced by the tree, particularly in the roots and or waterlogging. So that could signal the fruit, get ripe early so that it can have the seeds ready and things like that. And also when the trees are under stress, I think other responses may kick in. It's sort of, generally under stress situations, one response is the trees try to hurry up to finish the process, the seed producing process. But ethylene most likely plays a role in that.
Excellent. Thank you, Aaron. And Jamie, do you have any comments from your end?
A lot of the same challenges. But interesting that you brought up the micro sprinklers. So we do have a block of Honeycrisp where we have that, and we use it primarily for frost control in the spring, and that's been a concern we didn't anticipate, especially in springs where you have more frost events saturating that soil. This is on a relatively sandy farm. A lot of the western Michigan area is pretty heavy clay, and so we're usually more concerned about getting water away and not adding to it, but that's even a concern there. But I think that one of our bigger challenges, I think a few people alluded to, is we put in a lot of drip irrigation and have all these zones set up, and the zones were kind of set for what the well capacities could handle and not necessarily what soil types were or block setups were and things like that. And now we're realizing, like, we go turn the system on for an hour and we try to run all the zones to maximize the day, but we don't really have a good idea of how much water we're putting on or how much water that area needs. And so that's a lot of our work is focused on trying to dial that in, and like, some of the meters and things that were talked about, getting a better handle on that. Because once we know that information, then we can better utilize the models that are available, through NEWATER. For us, it's on EnviroWeather with the deficit irrigation and things like that. And to back up, I think maybe this might have been what Emily was talking about earlier, but so she mentioned last year, our spring anyway, after probably, June, July, before we had zero rain for a month. Zero. So people were doing what they could to try to irrigate and keep up. But what we did notice was that it was a really, really good year for Honeycrisp quality, especially where bitter pit was concerned, which seems to line up with all the numbers, because in that period that would be in that, 60, 80 days after bloom, we just had no more water. And then it did turn back on with rain, and irrigation supplement. But we definitely saw that reflected in the quality and throughout the storage period as well. So it was present at harvest, but we were able to keep storages that were pretty high.
Great. Thank you, Jamie. I just want to jump over to the chat and maybe pull Lailiang and Alan back into this, and the growers as well can comment with any of this. Lailiang, I'm just wondering if you wanted to expand any more. There was a question about the time of day for irrigation and whether there's any impacts with that. Do you have any additional comments to that? Oh, you're muted. Lailiang, I think you're muted.
Alan is in a better position to answer that question, but to me, time of the day doesn't matter much, particularly when you have a large acreage. There's no way you can irrigate all the trees at the same time, so you need to rotate anyway. And also, Alan, perhaps in a severe water stress situation, maybe irrigating the trees, let's say just before the lowest stem water potential, might help a bit there, but generally speaking, I don't think time of day would make a big difference.
Okay. Yeah, I think I agree with you about we can talk about being really precise as to when you irrigate, but in a lot of cases you don't have that choice.
Yeah.
You can't always do what you ideally want to do, but one of the things with I mentioned like apples have a very, very low amount of roots.
Root density, yeah.
Grasses or things like that. And so what happens in the middle of the day, even though there's far fewer roots than, say, grass, the water use of an orchard is fairly similar to that of grass.
Mm-hmm.
So you've got a lot fewer roots taking up the same amount of water. And so there's fairly good evidence of modeling and other things that the apple roots probably suck the water out of the soil just around the roots very quickly, in the middle of the day when it's really losing a lot of water. And so, in theory, it could make some sense if you wanted to kind of reduce that lowest value in the middle of the day, the most stress, to maybe put the water on. It would depend on your soil, but maybe like late morning, and then the water can get down to the roots right in the middle of the day when the water is the demand for that water is the highest.
Yeah.
So I think maybe something, especially if you're trying to grow young trees, I think with relatively few roots, I'm thinking that again, if you had the opportunity, it would be interesting to see if maybe like a late morning starting of the irrigation, those trees be able to maintain that vegetative growth to be able to fill the canopy. But
that's a good point, Alan. For young trees, and we noticed this too. I think we grew trees in sand culture, and if you irrigate, let's say late morning, just before noon, you get enough moisture there that can really prevent the large drop of midday stem water potential. I think that's a valid point.
So if you have heavier soils, it may take longer for that water
to percolate down. Yes.
So
could I ask a question on this subject?
Sure.
Okay. So we noticed we were using dendrometers over the last few seasons, and what we noticed was that if we irrigated at night, because there's always that expansion and then the fruit contracts and you lose a little bit, kind of like one of the graphs on the stress kind of showed, increase
a bit and then goes up. Yeah,
exactly. And we noticed that if we irrigated at night, it actually didn't go backwards as much than if we irrigated during the day. And so we kind of took the approach that, well, we thought for fruit sizing, that was kind of our goal, was going after fruit size. And so we were, all right, well, we started taking the approach to irrigate at night. Any thought on, when you start getting temperatures above 90, the tree's shutting down, is it, stomata are closing, is it taking up as much water during the day as it does at night under those extreme stress conditions at 100 plus? Does that make any sense, or that question, or that kind of line of reasoning? That's where we got to as well. It's cooler temperatures at night. It seems like the tree's taking up water better, and it was sizing the fruit and not going backwards as far as when we irrigated during the day. Does that make any sense, or is that yeah?
I think we lost Alan there.
Oh, we lost
We appear to have
Alan.
lost
Okay.
Yeah,
Alan.
that it was ready probably to give you an answer on that. I just want to add to that, Aaron, that one of the things that we have to consider is that transpiration happens also because you are doing photosynthesis, so that happens during the day. But there is that transpiration effect too that we have seen in Washington, especially, that the hottest time of the day start around that 4 p.m. and gets hotter and gets hot during just before the night. And so cooling the soil and cooling the environment also help reduce the respiration process, which will also help to keep the carbohydrates to increase the fruit size rather than to respire. So maybe there is something on that line that can justify your response on the practice that you are doing. I don't know, Lailiang.
Yeah, that's in the state of Washington, the environmental conditions are quite different from New York. So when I replied that message, in New York, really doesn't make much difference. But I can see the reasoning Aaron has, like irrigation at night or irrigating the trees, let's say in the afternoon. That can really cool down the orchard floor and helps lowering the temperature. And also that would help with the recovery of the stem water potential going into the night when fruit growth, it picks up. So, all of these, I think from that perspective, time of the day, may make some difference, especially under hot and dry conditions in Washington. I think, if you irrigate at night, because fruit growth typically, I hope Alan's back on, fruit growth sort of starts late in the afternoon, and then once get into the night, really accelerates. So that the water status there, we want to, if it's too dry, stem water potential is too low, they're going to limit fruit growth. So, from that perspective, sort of late afternoon, early night irrigation would also make sense. What Alan said was because the apple roots have very low density, under extreme stress conditions, evapotranspirative demand. So literally, the uptake of water by the roots creates localized drought around the roots. In that case, having water going down, making the roots in contact with the soil would be helpful too. So that's, sort of, I can see both reasoning, to me makes sense.
Lailiang, one thing I would like to share, at least we don't do like a cooling mechanism around here, but in the Hudson Valley, especially before a heat wave, some growers pre-irrigate to be able to prevent sunburn. So that is something that Hudson Valley growers, some of them do. In Hudson Valley, you have to irrigate. We have a significant amount of acreage in Western New York without irrigation. But the pre-irrigation before a huge heat wave is something that some growers are doing in the Hudson Valley.
Yeah, you basically load the soil with water. You just increase the soil water reserve. That's always a good idea before a heat wave. Yes, Mario, thank you for bringing that up. Yes, before
we are not doing that here in Western New York, but in Hudson Valley, I know some growers have been implementing that in the past.
Yeah.
Brian has the hand up, so.
Yeah. So one of the things we, when the batteries ran out, we stopped using our zone valves because I was finding that I was getting lazy. Once they were set and we turn the water on, the water comes on, right? And you don't worry about it. What I wasn't paying attention to something there is that we should have been cycling our blocks so that they were getting irrigated at night versus getting irrigated all the time at 3 o'clock in the afternoon. That block was probably getting maybe the wrong volume of water because it was at 3 o'clock in the afternoon all the time irrigation. Whereas if it had been irrigated at 9 o'clock at night, which I'm noticing is there is a definite difference. And then what we found is by irrigating that block at 9 o'clock at night, then actually having it on at 6 o'clock in the morning. So we irrigate from 9 till 11. And then I started again between 5 and 6, and I shut it off an hour later. And I find that soil stays has much better water capacity during the heat of the day than the block that if I would irrigated it every day at 3 o'clock. It's an amazing phenomenon to watch how the soils kind of react to that, that trend, that natural trend. So, yeah.
So, Brian, just want to make sure I understand you. So you irrigate, let's see, one block. You typically irrigate at 3 o'clock in the afternoon, right? So
well, you mentioned something, Dr. Cheng, is my farm's too big for me to there is no perfect time of the day to irrigate.
Yes.
Right? We have too big of zones or too small of zones or too many zones. So what I ended up doing now is we only irrigate zones by 2-hour blocks.
Okay.
Right? And so what we'll do is we cycle those blocks to different times of the day all the time. We never irrigate one block just at 3 o'clock in the afternoon till 5. We irrigate it we'll irrigate it at 9. We have a calendar. We'll irrigate from 9 to 11. When we turn on the pump in the morning, it's going to get irrigated for another hour. And then it's not going to get irrigated again, maybe till the next day, till maybe at 3 o'clock in the afternoon. Because we're trying to cycle that water overnight, allow it to soak in, and then and just keep those soil capillaries happy, and always pulling from. We want the soil to pull from the water table, not to be feeding the water table. We want the water table to work for us, right? So yeah.
So you notice that for the blocks that are irrigated from 9 to 11, the soils have better water status than those that you irrigated at, around 3 o'clock.
Yeah.
In the afternoon. Yeah. Yeah, that's 9 to 11. That's sort of before the midday stem water potential drops down. That's the sort of Alan talk about that. And then the state of Washington, mid-afternoon, late afternoon is a good timing. I think different timing of the day may cause sort of differences in the water loss from the soil surface as well. So that's another factor. It's a complicated.
There's a question that's been in the queue for a while. I'd like to get asked before we draw things to a close. We're already at 8:45. We could probably talk for another 2 hours, I'd guess. The question came from, we get the name back up here, is Leslie Huffman. It says, does Alan or Lailiang have any data on pear growth? Since Alan wasn't back on, I guess that falls into your camp there, Dr. Cheng.
Could you read the question again?
I will read it again because it was sent directly to me and you didn't see it.
Okay.
So it says, do you have any data on pear growth?
On pear? No, I don't have any. But I think Terrence might have some. In terms of growth patterns, apples, pears should be very similar. All this expolinear growth. But pear trees generally require, more water. And if you measure leaf photosynthesis, you're going to find that pear leaves have higher photosynthesis than apples, and the stomatic conductance is higher as well. So pears do need more water.
Okay, thank you for answering that. And one more here from Gil Costa. Are apples isohydric or anisohydric? And perhaps you might want to explain what those terms mean in case people in our audience don't know what
that's a term used for, in the tree world. But let me explain it this way. So we can contrast apples with the grapes. maybe that helps understand the term. For grapes, they have large, if you look at the xylem vessel in grape canes, you're going to find the xylem vessels have large diameters. Okay, so that really facilitates water transport from the roots to the above ground, to the canopy. So if you measure the stem water potential of grapes under well-watered conditions, the stem water potential of grapevines is always higher than apple trees. So for apple trees, their xylem vessel size, the diameter, is always smaller than grapes. So for herbaceous plants, they all have fairly large xylem vessels. So they have called higher hydraulic conductance. And for trees, for most of them, just like apples, they have lower hydraulic conductance due to smaller size xylem vessels. So maybe that's easier to understand than using those terms.
Okay, thank you. I believe that's all the questions from the chat. So I'm going to sign off here or shut down.
All right, thanks, Daniel. Yeah, I think with that, we'll wrap up the session and put some pins and a few questions. I know I have a few questions that we can kind of revisit next week. So if you would please take a few minutes and just respond to the survey Bernadita put in the chat, or you can use the QR code that came up on the screen. But I would really like to thank our speakers tonight, Dr. Cheng and Dr. Lakso, and all of the grower panelists for sharing your expertise and insights with this group. So thank you, everyone, and have a good night.